Well, it's a pleasure to be here to see so many old friends and meet new ones. It's just a great group of people, and sometimes I think I'd want to come here even if I wasn't doing any work on FSH. So yes, I've been tasked to talk about advances in clinical research, and the last time that I talked to this group was in Vegas in 2010. And prior to coming, I had done a search on clinicaltrials.gov, which you can also do. Just put in Facioscapulohumeral muscular dystrophy to see what studies or trials are being done. And they'll list both current and previous ones. And so in 2010, there were only 3 studies in FSH, and none of those were drug studies. So they included things like the the FSH registry. And you know, for many of us and those who are researchers and those who have FSH, that was just really appalling. And that's the reaction of Alan Brown, who is a friend, patient, and a clinical trial participant. And so I did this search again prior to coming to this meeting, and I'm sorry to say that there are only 5 studies listed on clinicaltrials.gov. Only one of those is a drug study, and that's in France and in the Netherlands. And at this point, Alan and I have just grown a lot of gray hairs and kind of dismayed over the situation. So I had talked to you about, and actually Amgen had talked to you also last time about myostatin inhibitors, and one myostatin inhibitor that we thought would progress to a clinical trial in FSH was based on the activin receptor. So it's abbreviated ActR2B, and it's the presumptive receptor of myostatin, which is an inhibitor of muscle growth. And so when you block myostatin, you get increased muscle regeneration and decreased scarring or fibrosis. And ActR2B does that extremely effectively in animals. But ActR2B also binds and inhibits other molecules similar to myostatin. So it's not specific, it is extremely potent. And we did begin to develop a plan for FSH. So there was a plan to do a clinical trial, and this was work that was developed in collaboration with the Wellstone Center and Acceleron. And unfortunately, that's been nixed. And there are probably multiple reasons why that has been nixed, and not all of which I probably am privy to. But anyway, it was an industry decision. And ActR2B was tried, or their drug ACE31 was tried in Duchenne muscular dystrophy. And that trial actually was terminated for safety reasons because boys developed nosebleeds and new blood vessel growth on their faces, which is just not acceptable, particularly in children. So it's possible that the FSH community didn't miss out on a whole lot there, but it was still really a disappointment for all. So what do I think are the potential things that are coming down the road? And this is my opinion and speculative. I think within the 2-year time frame, we're going to have what Fady Malik just mentioned, as relatively non-specific treatments to improve weakness. So that it does include, I'm very hopeful, and I can probably speak more candidly than Fady can, because he has to represent his company. But we're definitely hopeful that there will be a trial of CK. What are we calling it now? CK357, Maddie? Okay, in FSH. And there may still be a trial of a myostatin inhibitor in FSH. There are other companies that are pursuing that, and so I still think that would be a good thing. In the 5-year time frame, I think that some of the research that you heard yesterday may come into clinical trials. So you heard a lot about DUX4. You heard about silencing of DUX4 through RNAi, which is also sometimes called knockdown. And then there may be targeting of other misregulated proteins or genes in FSH, such as ones that the Wellstone has identified. And then 10 years down the road, I hope that there will be more cell-based therapies. And I think that area of muscular dystrophy research lags behind the pharmacological research, but it will be extremely important for all. So you've heard a lot about testing in animal models. So all of these potential therapies will go through these what we call preclinical testing in animal models. And you've heard already about a lot of the limitations of animal models. And one of the reasons that we don't have an animal model that everyone agrees upon is that we don't all agree upon or don't know what the molecular basis is. But even if we did, even if we said, okay, this one gene is the end-all and be-all of FSH, there's still a lot of limitations to animal models. So they have unclear translation to humans. For example, the only pharmacologic agent with a proven benefit in muscular dystrophy, prednisone, in Duchenne muscular dystrophy, developed not from animal studies, but from anecdotal observations in patients. And in fact, still doesn't have much of an effect on mice. And several other agents with promise in mice had no clear benefit in muscular dystrophy in clinical trials, such as creatine, glutamine, coenzyme Q10. And this isn't unique to the muscular dystrophy field, so there have been beneficial effects on the preclinical animal model of ALS called the SOD model that just really haven't translated into humans in terms of having a benefit. And the one exception to that is the drug riluzole, which is being used in humans with ALS. So you heard a bit about xenografting and how that might be one approach to this problem. And xenografting has been very helpful in other fields. So human tumor models are used in assessing if an individual patient's tumor will respond to a specific therapeutic regimen. And there have been successes in that regard. Multiple myeloma, for instance, showing that Velcade and also using that with other drugs, it can specifically improve multiple myeloma. But there are also limitations to this model also. It will always be, to more or less an extent, a hybrid of species. There'll be a little bit of mouse and a little bit of human. And meaningful measures of whether your therapy or drug is effective in this model are reduced compared to the whole animal. You may not be able to put it on a treadmill, for instance, and be able to determine whether the drug is efficacious if you're only affecting certain muscles. And then it's very labor intensive. You have to make each mouse as opposed to breeding them down in your colony. I think the next couple slides are graphic. If there are any kids perhaps who don't want to see cut open mice. Okay, so how we do this, there are many different ways to do this. I'm just going to talk about how my lab is doing this. We're actually transplanting a portion of FSH human muscle. So on the left, we take out the tibialis of the mouse. So this is the mouse knee and the mouse ankle. And then we sew in a piece of human muscle just right where that mouse muscle was, and we sew it into the tendon so it'll get the contraction. And then we sew the mouse back up. And this is an immunodeficient mouse. It doesn't have an immune system to respond to noticing that it's not mouse, that it's human. And surprisingly, the grafts take extremely well. This is after 20 weeks. You can see that the muscle is pink. and healthy, and then we can do studies on it. And the human muscle that this came from are some of you in the audience. This is through the biopsy program that the FSH Society helps coordinate, and the Wellstone helps fund, well, the Wellstone funds. So this is what the graft looks under the microscope. Okay, so this area here up to about this line is all human, and each of those polygonal shapes is a single muscle fiber. And how do I know that's human? We can put on an antibody that specifically recognizes human, and the antibody is only recognizing this portion and not this portion, which is mouse. Dr. Emerson mentioned yesterday that he had wanted to show innervation of the xenografts. So in A, that is a picture of antibodies recognizing the mouse nerve, a mouse nerve coming into the graft, and B, that's an antibody recognizing a protein on the muscle called the postsynaptic membrane, and C shows how those two are meeting. So you have a mouse nerve innervating the human muscle, and that's good. That's important, because otherwise you've got a model of something else. You maybe have a model of atrophy due to loss of nerve, but we believe this is a decent model to try some of these new drugs. And then the question was raised yesterday, well, what do these really look like, these xenografts? And they're actually they're very cute. I've thought about taking one home. So Fatty has already talked to you quite a bit about drug development, so I'm not going to dwell on that. But I'd like to talk to you a little bit about what your role may be moving forward, what you might be asked to do. So in clinical trials, we actually start with the preclinical trials, and then a drug goes through something called phase 1, where a limited number of subjects who are usually healthy, who usually don't have the disease, FSH in this situation, will be tested for the drug. And that's going to be to look at safety measures, toxicity, but also to determine how long does the drug last in your bloodstream, in your body, for instance, try to determine how often it should be dosed. And so they may get the drug once, or they may get it a few times, and there may be different parts to a phase 1 trial. And then a phase 2 trial is where you continue to look at safety, but now you're looking at safety in the disease population, and you're beginning to look at efficacy. And again, there may be different phases of the phase 2. And then finally, a phase 3 trial would have quite a bit more subjects involved. And again, this is to really validate the efficacy of a drug. There are some important concepts to a clinical trial that one should know. Participating most clinical trials are placebo-controlled, and that means that some participants get a dummy. And the placebo-controlled is really critical, not only for understanding the efficacy, but understanding the safety of a drug. So without that group of controls, then anything, for instance, all the patients had diarrhea, constipation, you know how that's listed on every drug, that all gets ascribed to the drug. Clinical trials, we hope, are randomized. So the subjects are randomly assigned to a placebo or dose group. You know, if you're my patient and I'm running the trial, I don't get to put you in the drug part as opposed to the placebo. It's randomized. It's double-blinded, so neither the investigator nor the subject knows what group or arm he or she is assigned to. And very frequently, specifically for phase 2 studies, that there are very strict what are called eligibility criteria. And the purpose of this is to try to get a homogeneous, as close as possible group of subjects. So I could imagine with FSH that, we might be really excited about a troponin activator, but we're not going to want to try to test that on, for instance, all 3 subjects who were up here previously. People. So, we're going to want to take one type, and with one type of severity, and get a group of those people who are all very similar to see whether the drug works. So we're not comparing apples and oranges. Many clinical trials are randomized control trials, and now you know what RCT means, are parallel group designs. And this sort of shows you a parallel group design. We get some baseline information. We randomize people so they either go to treatment A or to treatment B, and we usually study them over a long period of time, relatively. And it can have multiple arms. It doesn't have to have 2, and they don't have to have equal numbers in each arm. You could have, 3 people receiving the treatment and 1 for every 1 receiving placebo. Another possibility, however, is a crossover design. In this design, subjects receive different treatments during different time periods, and each individual serves as their own control. For this type of design, the disease has to be chronic and relatively stable, and FSH usually is in that regard. Over a 1-year period of time, many of you may feel that your disease doesn't necessarily change much. Over a 5-year period of time, it does. These types of trials need to be relatively short, and you have to have a washout period. So you receive a treatment and then it washes out, and then there can't be a carryover effect from having received the previous treatment. And this type of design would fail if there's a high dropout rate. People start, we get some baseline information, they get a treatment, there might be a washout period, and then they might be on a different treatment, and that might be control and then treatment. And this is a possibility of a trial design that wouldn't cure the disease, but might make somebody stronger, like maybe the troponin activator, ZK357. So there are some negatives of clinical trial participation. There's risk. There's risk of the drug, and I think that's exemplified by the recent ACE-31 trial. There's risk to the procedures. Many clinical trials, such as the myo-29 myostatin inhibitor for FSH, had multiple muscle biopsies, and there's risks to that. There's risks to your privacy. I think the biggest risk is inconvenience. It's very frequently a big time commitment, and travel may be involved. There may be expense. There shouldn't be any expense in terms of participation in the trial itself. Any of the procedures, for instance, are always covered by the medical institution or the company, but there's, you may have unanticipated or personal expenses. You may have to miss work that you're not compensated for. There's discomfort frequently, and as many in the myo-29 trial will attest, there's disappointment. The drug may fail. It usually does. Most drug trials fail in terms of showing an effect, and industry will not necessarily pursue it. You may think this was a great drug, and I personally had a benefit to it, but it may not, for a whole group, show a positive enough effect to be pursued. There are also positives to participation in clinical trials. I think one of the reasons that people participate is the potential for earlier access to an efficacious drug. That doesn't always happen, but many industry sponsors say that if you're, that we can't pay you for participating and undergoing all those discomfort, expense, and convenience, but if we are pursuing this, then we'll have you on the drug in an extension trial while we pursue FDA approval. You get a lot of medical attention, and maybe that's not all positive, but but sometimes it is. Sometimes by this, extraordinary, frequent visits and number of tests, you pick up things that would otherwise have been overlooked, or you learn things, or we say, after seeing you week after week, we say, have you ever thought about this piece of equipment that you just wouldn't otherwise have had the opportunity? I think one of the big pluses is the empowerment that you feel of personally moving the field forward. I think a lot of people feel that way from fundraising and contributing to moving the FSH Society or research forward, but also participating in clinical trials. You are an active participant moving things forward, and we really do move things forward. So the community learns about FSH. We learn about the natural history, and that was one of the positives of the Myo 29 trial, that we have a lot of data on patients over a year of time, and for the placebo and the low dose groups, we've gained a lot of information there. And then the community learns how to study FSH with biomarkers, imaging, endpoints, et cetera. And you've just really heard very candidly from Fatty that they picked ALS to do a clinical trial because there was experience in ALS and there were endpoints, and that's discouraging to us in that FSH is just as prevalent as ALS is. There are no treatments for FSH, and there are no treatments for ALS, really. I mean, in many ways, they're very equivalent. So it's discouraging, but on the other hand, the lesson to be learned is that we have to do clinical trials in order to get the drug companies interested to get better treatments and cures. So, the rich get richer. The more we do this, the better we'll be. And I'd like to just end with a quote. This is Don Burke, and he participated in the Myo 29 trial. And I'd actually just like to recommend all of the personal observations that were published in the Watch newsletter, which I think is just really fantastic, from research and patient perspectives. But many of the participants in the Myo 29 talked about their experiences. What year was that? Okay, 2007, spring 2007. And anyway, this is Don's quote. To my surprise and appreciation, I found myself immersed in a medical system that overflowed with passion for its work and for FSHD. I was surrounded by doctors, surgeons, nurses, and aides who were attentive and engaged. I found myself looking forward to each visit and the exchange of information and knowledge. And so I hope that everyone who participates in an FSH clinical trial will have such a positive experience, and I personally will work to make sure that that happens. So I'd like to acknowledge my research staff, and in particular Tracy Zhang is here. She is doing the Xenograft work, and I welcome you to talk to her and to volunteer. And my clinical staff, which really makes it a pleasant experience to visit Kennedy Krieger, that's outside of our outpatient building. We have funding from the FSH Society to do MRI studies, and are currently recruiting to do MRI of skeletal muscle. And that's my email address and my telephone numbers. I didn't bring business cards, unfortunately, but I welcome any of you to contact me. Thank you.